Good morning to all. We now move to the second session of this workshop, but firstly I would like to acknowledge the organization for inviting me to participate in this interesting workshop. This thematic is indeed very pertinent presently, and the workshop addresses lots of topics that are related with the anti-cancer drugs. And this session will tackle the environmental monitoring and toxicity assessments of the chemicals that are used in the treatment of this type of diseases. And I would like to highlight you that we will have firstly the 4 talks, and we will have only at the end of the session 10 minutes for discussion. So you have 2 options that you can use to raise or to put your questions. You can write them in the chat, for example, during the presentations. You can leave your comments or your questions in the chat, and then we can go through them during the 10-minute session. Or you can do as we have been doing until now, just by raising your hands. Okay, so let's move to the first talk of this session, that is entitled Anti-Cancer Drugs in the Environment, Occurrence, Risk Assessment and Degradation, by Teresa Gouveia. In brief, Teresa Gouveia is a PhD student at the Environmental Science and Technology Research Group of LEPABE from the Faculty of Engineering at the University of Porto. The research work of Teresa is focused on the development of cost-effective approaches for the mitigation of risky cytostatics in Portuguese waters, and her talk will summarize the results that she is obtaining in the research team where she works on the levels of cytostatics in wastewaters and surface waters as well, and the technologies for cytostatics removal degradation. So Teresa, welcome. You may start your presentation.
So hello everyone, my name is Teresa Gouveia. I'm a PhD student at LEPABE, FEUP, Faculty of Engineering of Porto. First of all, I would like to thank the organization for inviting me to be here presenting the work my group has been doing. My supervisors are Dr. Mónica Santos and Professor Arminda Alves, and the theme of my presentation is Anti-Cancer Drugs in the Environment, Occurrence, Risk Assessment and Degradation. I would like to start making a brief introduction to the topic, focused on cancer incidence and the use of anti-cancer drugs in chemotherapy. Then I will talk about the anti-cancer drugs of most concern worldwide. Afterwards, I will focus specifically in the case of Portugal, starting by the consumption trends and the predicted concentrations of these drugs in surface waters. Then I will talk about a study we did where we measured the concentrations of anti-cancer drugs in the inlet and outlet of a Portuguese wastewater treatment plant. With these concentrations, we estimated their risk in the surface waters and estimated the risk quotient and determined if aquatic organisms could be or not at risk. Finally, the efficacy of the degradation treatments in the elimination of anti-cancer drugs in that wastewater treatment plant was evaluated. As we all know, cancer is nowadays one of the leading causes of mortality worldwide. In this map, we can quickly check the estimated crude incidence rates of all cancers in 2018 across the globe. The countries highlighted in dark blue are the ones for which a higher cancer incidence was recorded in 2018, and Portugal is among these countries. The forecasts of the International Agency for Research on Cancer pointed out that the cancer incidence will increase approximately 40% from 2080 to 2040. With the increase in cancer incidence, there is also an increase in the administration of anti-cancer drugs. Upon administration, anti-cancer drugs are excreted by human body and end up in sewage system. Since wastewater treatment plants are not effectively degrading these hazardous compounds, they are getting into surface waters and the overall environment. In hospital effluent, concentrations up to 123 micrograms per liter were detected for fluorosil. In the entrance of a wastewater treatment plant, the maximum registered value worldwide was 13.2 micrograms per liter for cyclophosphamide. A concentration of 1,200 nanograms per liter was found for carboplatin in a wastewater treatment plant effluent. And after leaving the wastewater treatment plants, anti-cancer drugs reached the rivers. Mycophenolic acid was detected in concentrations up to 556 nanograms per liter in a river, which is very high for that metric. After treated, the water is available for consumption. Although most of the studies are not able to detect anti-cancer drugs in potable water due to the method detection limits that they have, bleomycin was already detected in a concentration of 13 nanograms per liter, which is very high in the water that we drink almost every day. Now I will present a quick study where we define the anti-cancer drugs of most concerned worldwide. So the risk quotient should be determined based on measured concentrations whenever possible. A literature review on concentrations of anti-cancer drugs measured in surface waters worldwide was performed until 31st of December 2018. The risk for each anti-cancer drug was then determined for the highest concentration measured in surface waters worldwide, and also for the lowest method detection limit reported in the monitoring studies, since all anti-cancer drugs were not detected at least once in every study. The risk quotient is then calculated by the quotient between a concentration and the predicted no-effect concentration. The predicted no-effect concentration were defined as the lowest values obtained from the quotient between the toxicity data for each cytostatics and an assessment factor, depending on the toxicological dose descriptor and the number of non-trophic toxicological levels. The risk quotient is higher when we have high concentrations or high toxicity that leads to lower PNECs, predicted no-effect concentrations. The anti-cancer drugs that have been studied in surface waters are compiled in this table. Analyzing the second column, which represents the risk quotient obtained from the maximum measured concentrations in surface waters worldwide, we can conclude that mycophenolic acid, tamoxifen, and fluorosil may be posing aquatic biota at risk at the concentrations they are measured in rivers. Methotrexate and tegafur a moderate risk, and bicallutamide a low risk. We defined as a high risk when the risk quotient is higher than 1, moderate risk when it is between 0.1 and 1, and a low risk when it is between 0.01 and 0.1. Looking to the third column, which represents the risk quotients obtained from the lowest method detection limit reported in monitoring studies, we can conclude that the available analytical methodologies for the analysis of cisplatin, daunorubicin, fluorosil, imatinib, and also mycophenolic acid do not allow to rule out the existence of risk. This means that these compounds may be posing aquatic biota at risk even though they were not detected. This also highlights the need for more improvements in the sensitivity of the analytical methods to detect these compounds. So now the question is Portugal following the same trend? Now I will talk about the consumption of anti-cancer drugs worldwide. Even between different regions of Portugal, differences on consumptions were recorded. The center of Portugal registered the highest consumption per inhabitant. This data was collected between 2007 and 2015. The lowest consumption records were observed in Alentejo and Algarve. However, according to the national report, Center was the region with the lowest cancer incidence in 2010, which seems not to be in line with this trend. So this may be a consequence of the location of the National Oncological Institutes in the North, Center, and Lisbon regions. Moreover, it may reflect the presence of patients for certain hospitals located in these regions to treat some cancer pathologies, even though they were outside the residence region. We can also conclude that the most consumed anticancer drugs in Portugal are mycophenolic acid, hydroxycarbamide, capecitabine, fluorouracil, and bicalutamide. Up to now, there is no information about the occurrence of anti-cancer drugs in Portuguese surface waters. So we decided to estimate those concentrations based on consumption records. To estimate the concentrations of anti-cancer drugs in surface waters based on consumption records, this equation was applied. Beyond the consumption of anti-cancer drugs, other parameters were considered: the consumption of water per inhabitant and the number of inhabitants which were obtained from national ministries. The percentage of excretion of the parent anti-cancer drug, the removal efficiency at wastewater treatment plants, and the dilution into receiving waters were also part of this equation. Whenever possible, experimental data was used for the excretion factor and the removal factor. If more than one value was available for the same parameter, the one associated to the worst case scenario was selected. Concerning the dilution factor, the value of 10 was proposed by European medicine agencies, and it was the value that we used. So, the anti-cancer drugs whose predicted concentrations in surface waters were higher than 1 nanograms per liter are depicted here in this graph. Mycophenolic acid was by far the anti-cancer drug with the highest concentration in surface waters, followed by hydroxycarbamide. In the other study that I mentioned, the worldwide study, we could also see that mycophenolic acid was the one present at higher concentrations in rivers. Contrarily, cyclophosphamide, ifosfamide, and imetinib are probably at much lower concentrations. So the question of concern now is, are these concentrations potentially posing aquatic organisms at risk or not? Once again, the risk quotient was calculated, in this case based on predicted concentrations. So the predicted no effect concentrations used in this assay were estimated for all of them from short-term toxicological data, using a security factor of 1000 to ensure that the compounds with the potential to cause adverse effects are identified in this effects assessment. So mycophenolic acid is suspected to pose high risk to aquatic biota, once again, as we have seen in the worldwide study. Although cyclophosphamide was one of the compounds with low expected concentrations in surface waters, a moderate risk is associated to its presence in the aquatic environment, which means it is very toxic. Bicalutamide show a low risk to aquatic organisms. Even though no evidences of risk exist yet for other anti-cancer drugs, concerns may be associated with long-term effects or synergetic effects. Now that we know the anti-cancer drugs that are probably posing aquatic biota at risk based on predicted concentrations, we decided to measure the concentrations these compounds are released from a Portuguese wastewater treatment plant and have a more accurate approximation of the risk. In this work, we collected samples from the effluents, secondary effluents, and tertiary effluents from a Portuguese wastewater treatment plant located in the north. The extraction process used was a liquid-liquid extraction, and the quantification method, a liquid chromatography mass spectrometry. There are some compounds that were not detected even in the effluents: cyproteron, flutamide, and paclitaxel. Other compounds were detected even in tertiary effluents, such as bicalutamide, capecitabine, cyclophosphamide, ifosfamide, and mycophenolic acid. All of them were found in most of the samples analyzed, at least 10 of 14 samples for each anti-cancer drug. Mycophenolic acid was also the anti-cancer drug found at higher concentrations in every metric study. These results are in accordance with the predicted ones that we did before. For the analysis of the risk these compounds may be posing to aquatic life, an estimation of their concentrations in surface waters was done. According to European Medicines Agency, a dilution factor of 10 was applied from the tertiary effluent to surface waters. And interpreting these results, we can conclude that mycophenolic acid is once again probably posing aquatic life at risk. The other anti-cancer drugs that were detected in tertiary effluents did not show any risk to aquatic biota because their concentrations or their toxicities were not high enough. The results obtained in this work can also provide us some highlights about the efficacy of the degradation of anti-cancer drugs in that wastewater treatment plant. Looking at the same table in a different perspective, we can see that there are some anti-cancer drugs such as doxorubicin, etoposide, imetinib, megestrol, mecophenolate mofetil, and prednisone that were quantified in the effluents but not in secondary effluents. This may indicate us that primary and secondary treatments are effective for the removal of these drugs to a level that is below our method detection limits, at least. On the other side, there are some compounds that were barely degraded, such as bicalutamide, capecitabine, cyclophosphamide, ifosfamide, and mycophenolic acid. If we look at the case of ifosfamide, it was not degraded at all. It even increased a little bit of the concentration, maybe due to the cleaner extracts of the tertiary effluents. So this means that these compounds are very, very recalcitrant to UV radiation. That is the treatment that is used in the tertiary treatment in this wastewater treatment plant. So the main conclusions of this work is that when we look at the concentrations that were measured in worldwide surface waters, we can see that a high risk was estimated for mycophenolic acid, tamoxifen, and fluorosil. Methotrexate, tegafur, and bicalutamide also indicates some risk to aquatic life. The first study can also tell us that the methodologies currently available for the analysis of some anti-cancer drugs do not allow the assessing of whether or not the aquatic organisms are at risk of exposure, because we calculated the risk based on the lowest method detection limits, and we found the risk for those compounds. So there is a need for the improvement on the analytical methodologies to detect these compounds, and then we can see if they are probably posing or not aquatic organisms in the aquatic environment. In the case of Portugal, the consumption trends tell us that the 5 most consumed anti-cancer drugs between 2007 and 2015 were mycophenolic acid, hydroxycarbamide, capecitabine, fluorosil, and bicalutamide. We could also conclude that the center of Portugal has the highest consumption of anti-cancer drugs per inhabitant, and Alentejo and Algarve the lowest. And we could also see that these values are not in accordance with the highest incidence of cancer. It is only those are only the regions that are providing more of these treatments. The predicted concentrations in surface waters and potential risks to aquatic organisms based on these concentrations told us that mycophenolic acid is the anti-cancer drug with the highest estimated concentrations in surface waters, followed by hydroxycarbamide. Cyclophosphamide, ifosfamide, and imetinib are expected to be at less abundant anti-cancer drugs in Portugal. However, cyclophosphamide, as we've seen, although it is present in low concentrations, it was estimated a potential risk associated to this drug. Based on the measured concentrations in a wastewater treatment plant, we detected that mycophenolic acid was once again the anti-cancer drug present at higher concentrations in the effluents, in secondary effluents, and also in tertiary effluents. Bicalutamide, capecitabine, cyclophosphamide, ifosfamide, and mycophenolic acid were detected in tertiary effluents in most of the samples tested. And we could also conclude that mycophenolic acid is potentially posing aquatic organisms at risk, as we have seen in the other 2 studies. We could also conclude that in the study that we did in the Portuguese wastewater treatment plant, doxorubicin, etoposide, imetinib, megestrol, and mycophenolate mofetil and prednisone showed to be degraded in primary and secondary treatments. We detected them in significant concentrations in the effluent, but then they were not detected in the effluent, which is good. However, bicalutamide, capecitabine, cyclophosphamide, ifosfamide, and mycophenolic acid showed to be very recalcitrant to UV radiation. Cyproteron, flutamide, and paclitaxel were not detected in any sample. this work tells us that there are some compounds that are probably posing aquatic organisms at risk, but not only aquatic organisms, because then the water is treated and we drink that water. So there is a need for studying more degradation treatments to improve the degradation of these compounds, especially in wastewater treatment plants. We could apply these treatments in the hospitals, for example, but there are some compounds that are administered at homes. So the focus of the following studies is to study more degradation treatments to apply in the wastewater treatment plants and to prevent these compounds to be released to the environment. I want to thank everyone that helped me do this job, and you all for the attention.
Thank you, Teresa, for this nice talk. We can move now to the third part of this session. We will have Cátia Venâncio talking about the ecotoxicological tools to assess the adverse effects of cytostatics in freshwater environments. Cátia Venâncio, she is a junior researcher at the Department of Life Sciences of the University of Coimbra, where she develops research mainly within the area of aquatic ecotoxicology, so trying to understand the adverse effects that the diverse chemicals that are produced by or that are produced anthropogenically may cause in the biota, mainly in freshwater organisms. Cátia has been involved also in this project, in the IonCytDevice, and she has been collaborating specifically in this research subject, so she is trying to characterize the toxicity of some cytostatics to freshwater biota, and also she will be handling in the future the characterization of the toxicity of these samples that Francisco just talked about after being treated with these supportive ionic liquids. So, Cátia, thank you very much for your availability to give this talk. You may start.
Hello, good morning to you all. My name is Cátia. As Isabel said, I'm a collaborator in CESAM at the University of Aveiro. First of all, I would like to thank the organizing committee for this invitation to participate. And I come to tell you a little about the ecotoxicological tools for the evaluation of the effects of cytostatic drugs in the aquatic environments. I will also want to say that this work has been developed mainly within the scope of a master thesis of Bruna Monteiro under the supervision of Dr. Ana Catarina Souza and Dr. Isabel Lopes. As said before, cytostatic drugs, also known as antineoplastic drugs, are drugs or pharmaceuticals of synthetic or natural origin, widely used in the treatment of cancer. In a very simple way, cytostatics block the replication of the DNA from the cancer cells and therefore prevent its proliferation and tumor formation. As said in the presentation of the workshop, statistics have shown that cancer remains a serious public health problem, and the number of cancer cases from year to year has been rising. For instance, in Portugal, in 2018, about 2% of the population was affected by some type of cancer, with 58,000 new cases reported in that year. In Europe, it was estimated that 17% of the population was also affected by some type of cancer, and worldwide, it is expected that more than 27 million cases will appear until 2040. Taking into account these numbers of cases that increase from year to year and on a global scale, the World Health Organization has already also reported a significant increase in the consumption of cytostatic drugs, and these numbers are expected to increase. But then after the administration of these drugs, what is their destiny, their environmental fate. So this scheme serves to exemplify how these drugs reach the environmental matrices. The main sources, or the key emission sources, as also said by Francisca, are hospitals, cities, and of course the pharmaceutical industries that produce these pharmaceuticals. In a study by Ort et al. From 2010, the authors found out that hospital wastewater would be only responsible for 7.5% of these wastes, of the release of these wastes, with the largest portion attributed to domestic sewage. But other recent studies also point to this type of statistics. And it must be said that wastewater transport not only secondary metabolites that result from the metabolization of these drugs by the patients, but also a portion of the cytostatics that have not been metabolized by the patient. These compounds will circulate through the sewage networks, and they will reach wastewater treatment plants. And here, in these wastewater treatment plants, also highlighted by Francisca, what happens is that they are not efficiently removed, either by limitations of the degradation processes or by the continuous release of these compounds into the sewer networks, which makes their total removal almost impossible. Okay. It should also be beared in mind that the interaction phenomena between these compounds and other compounds present in the wastewater might also occur, as well as with other matrices such as sludges. So, it is believed this way that aquatic ecosystems will be a major recipient of these drugs, which raises concerns among drinking water regulators, governments, water providers, and of course the general public about the proven and potential risks to human and environmental health. Also, although there are some studies focusing on the environmental effects of cytostatics, the truth is that their effects on non-target species in the environment are largely unknown. For instance, about their potential for bioaccumulation. This means whether or not they can accumulate inside living tissues. For instance, concerning their persistence, this is how degradable they are, whether it is abiotic or biotic degradation, for instance, photolysis, hydrolysis, or microorganisms activity. There is also a possibility of mixing effects, because these substances will not appear isolated in the environment, so they can interact with each other and with other present in the environment, and even result in synergistic effects. That is, the effect of the mixture is greater than the sum of the effect of each compound individually. Thus, the environmental load in the aquatic matrices is not well understood, because no technique can be applied or has been found to be able to remove all relevant pollutants from wastewater, and also because it is impossible to assess on a daily basis the environmental load of these compounds or their metabolites, and also because the research focuses mainly on those of more intense or popular use, like cyclophosphamide. Furthermore, also as said by Francisca, these drugs are designed to have an effect on patients in very small dosage. So even minimal concentrations can present a potential danger to the environment. And not being selective, they can act upon every eukaryotic cell, interfering with its DNA. There are several studies pointing to the cytotoxic, genotoxic, mutagenic, and teratogenic potential of these drugs. So, taking into account what was previously said, this study aimed above all, to gather from the literature data about the effects caused by 3 of these cytostatics, and to give answer to the evaluation of the efficiency of a new methodology to remove cytostatic drugs from the urine of oncologic patients. These are the 3 cytostatics we are working with: cyclophosphamide, 5-fluorouracil, and MMF. The first 2 are in the top 5 of the most monitored cytostatics in the environment. Talking about a little bit of each. Cyclophosphamide is one of the most consumed cytostatics, although very recent studies indicate that its present in its threat to the environment is virtually 0. Cyclophosphamide is considered persistent, and due to its high consumption rate, it is believed that it is continuously released into the environment. It is one of the cytostatic drugs for which more information about its ecotoxicity exists. 5-fluorouracil is also one of the most prescribed drugs for the treatment of cancer, and also for which more environmental data exists. It is considered as a pseudo-persistent compound. This concept is very important because although it is a compound that has relative short half-life and its degradation is possible, mainly due to abiotic factors, its continuous use and replacement in the environment makes it continually present. Hence, it is considered pseudo-persistence, reason for which it is considered a high-risk cytostatic for the aquatic environment. And MMF, it starts to be also one of the most explored and used cytostatics for the treatment of cancer, and also with a short half-life. However, its ecotoxicity is largely unexplored, but the few existing data suggest that it presents high risks to the environment. Despite this information that can be collected from the literature, the truth is that in terms of environmental effects, the information is still very incipient. It is essential that in order to have regulatory and risk assessment structures for these compounds, chronic or long-term effects, potential bioaccumulation, potential interaction with other compounds must be studied. Let's see with the example cytostatics what happens when we try to derive environmental safety values from effects values reported in the literature. Therefore, for the three cytostatics in the study, we collected from the literature average lethal and sublethal effect values, and we constructed this type of curves. Okay, this is called the central tendency. This line in orange, and it's accompanied by two other lines that are the confidence limits at 95%. Okay, and very quickly in this type of graphic, species are distributed along the curve from bottom to top, from the most sensitive to the most tolerant species. This type of analysis also allows us to calculate a safety value for the protection of 95% of the species in an ecosystem. It is called the hazard concentration at 5%, and after the application of a safety factor, it serves to derive, for example, the predicted non-observable effect concentration that I believe Teresa also mentioned, that are commonly used by regulatory or environmental risk assessment entities such as ECHA or REACH. So from this curve for cyclophosphamide, we obtained an HC5 value of 9.9 with a confidence limit that ranges from 1.66 to 63. And even if we apply a safety factor of 10, we will reach a value of 0.99 milligrams per liter. That, if we compare with the values of PNEC reported in the literature, you will see that is within the range. I selected these two from all the values that I found. I selected these two, the lowest one and the highest one, and you will see that it fits within. Okay. However, it might not always seem so linear. Let's look at the case of 5-fluorouracil. Okay, we did the same type of approach with 5-fluorouracil, and what we saw was that the central tendency curve, the line, this line, okay, for example, in relation to the previous one, as a part on the negative axis of the x. And what happened when we tried to derive the HC5 value? Okay, we got a value of 0. And why did this happen? This happened because the less data there is in the literature, the less robust the curve will be, as well as the parameters that we determined from it. Okay. Furthermore, the robustness of the curve also decreases when, if you look at the values that are on the bottom and the top of the curve. Which means, what do I mean by this? The average effect of the value for the most sensitive species at the bottom of the curve, comparatively to the average effect value for the most tolerant species, okay, the difference is over 3,000 times. Therefore, trying to derive or predict effects on the environment based on these values becomes a very difficult and compromising task. Regarding mycophenolate mofetil, or MMF, what happened is when searching for average lethal or sublethal values, we just came across these two values. It is impossible to construct a curve with only two values. Okay. There are, I must say that there are other values in the literature, namely NOx and low X, that is the concentration of non-observable effect, or the lowest observable effect concentration. Okay, but however, their use is discouraged because it depends mostly on the size of your sample in the N, also on the concentrations that you use, namely on the dilution factor used between the concentrations, and also depends on the variability of your data set. Therefore, taking into account that the prediction of effects at the environmental level, as well as the determination of safety environmental concentrations fails or it's still very incipient, and taking the presentation scheme of the workshop program, we, in the part of the ecotoxicology field, we try to improve the assessment of toxicity environmental risk of these cytostatics because of the lack of ecotoxicity data, as shown by the curves before, the SST curves, and we also can provide answers or clues in the development of remediation processes. So this second part, as the presentation of Francisca was devoted to this, this second part of the remediation processes involves the development of devices capable of retaining non-metabolized cytostatics or their metabolites from patients' urine, whether in the hospital or at home, in order to reduce their discharge into wastewater. Therefore, our part is to evaluate, for instance, the toxicity of the compounds in artificial urine before it passes through the device of the cytostatic retention, the devices for cytostatic retention, and then we will re-evaluate the ecotoxicity of the urine after passing through the device. What happens if ecotoxicity decreases? Okay, we are responding to what was intended, creating a device capable of retaining these compounds upstream, that is, in a hospital or a context home, before reaching the treatment plants. If the toxicity is higher or it remains similar, it implies that the structure of the device must be rethought. It is a dynamic process and it's currently being processed. Just to conclude, and also for you to understand how this ecotoxicity assessment process is carried out, ecotoxicity assessment involves the assessment of lethal or sublethal effects using representative species of different trophic levels. For instance, we have algae that represent the producers, okay, and in which we can evaluate effects on their growth or even chlorophylls. Rotifers that are from the same ecological group as daphnids, okay, and where we can evaluate mortality or reproduction. And for instance, vertebrate species, that is Danurerio or zebrafish, where we can evaluate, where we can follow the development of the egg until the larval stage, as you can see the picture from the right to the left. And we can evaluate a series of endpoints like survival, etching, and malformations on the tail, formation of edemas on the heart, etc. So I finished my presentation by thank you all, and thank you for for listening.
Thank you, Cátia, for your nice presentation. Let's move on to the last talk of this session. It will tackle the occupational exposure to antineoplastic drugs regarding the Portuguese reality and the way forward by Susana Viegas. Just some few notes on Susana. She's an assistant professor at the Nova University of Lisbon, so at the National School of Public Health. Her main research topics include occupational and environmental toxicology, exposure assessment, biomonitoring, exposure and risk assessment of mixtures and food safety. She is currently involved in a European project that aims the protection of workers from exposure to hazardous medicinal products, including cytostatics, medical product. And her presentation will describe the exposure of healthcare workers from Portugal to antineoplastic drugs. So thank you very much, Susana, for your availability to be here with us. Today is a very working day for you.
Yes, indeed. Thank you, Isabel, for the nice introduction. And sorry for this delay, this small delay. I will now starting my presentation. Hi, everyone. So thank you so much. So I will start to say thank you to the organizers of this webinar for the invitation, and thank you so much, and to congratulate also for the initiative and to keep going even with this pandemic, and of course to make a very interesting program. So I will present the main findings that we have in Portugal regarding occupational exposure to antineoplastic drugs. We start doing this study in 2010. And we focus on this exposure because we have several reports saying that the use of antineoplastic drugs was increasing in different settings. It was, of course, already known that they have some side effects, carcinogenic, mutagenic, and teratogenic effects. And since '79, since the 80s, it was also recognized that they are a potential health hazard concerning specific occupational settings that we are going to discuss it later. So several studies that demonstrate that nurses and also pharmacy personnel involved in the preparation or administration are exposed to antineoplastic drugs. Indeed, these two groups of workers are the most studied, mainly because they are working in an occupational setting that are the hospitals, where there are a higher number of these kind of treatments, but also because they are the ones that we know better, implicated in the preparation of the drugs, but also in the administration of the drugs. So how exposure can occur? Well, in the beginning, the exposure was occurring mainly by inhalation because most of the hospitals and the health service don't have specific dispositives to control and to cap the emissions when the drugs were being prepared. But nowadays, most of the hospitals don't have the perfect conditions, and most of them have already a cabin where the preparation of drugs are made. So in that way, they can avoid the exposure by inhalation. Exposure by inhalation can happen if there is an accident and something goes to the floor, and then during the cleaning procedure, sometimes it can happen also by inhalation. But nowadays, the most common exposure route is dermal absorption. And this happens because workers have to handle and touch several surfaces that are sometimes contaminated by antineoplastic drugs. Of course, the contamination levels should be as low as possible. We know that zero contamination is almost impossible. That is being mentioned by most of hospitals' service, but this is for what we should work. As we know, exposure can result in health effects, and it's not so much related with the dose. Simple contact, in theory, can result in a health effect. Surface contamination assessment is a common way to estimate occupational exposure, and the most common method used is wipe sampling. So we are studying surface contamination to estimate the exposure of workers when they have to be in the workplace context where there are surfaces that might be contaminated. So we try to understand what are the number of hospitals that handle antineoplastic drugs. So they need to prepare and administer antineoplastic drugs. So we use a report from Health Directorate from 2002, where in that report mentioned that 78 hospitals treat patients with cancer. So this is, in that date, was 82% of the total of hospitals in Portugal. From these 78, some have chemotherapeutic treatments, but it was not mentioned the number of hospitals that deliver this kind of treatments. I don't know if there is available nowadays other kind of reports. I was trying to look for something more recent, but it was not available. So the number of hospitals that handle chemotherapeutic treatment is very difficult to anticipate. So 50% of the hospitals administered more than 2,165 treatments. One hospital with a great number of treatments, probably an hospital linked with this kind of specific therapies, of course, dedicated to cancer treatment. 4 hospitals with a similar number, 11 hospitals between 3,000 and 7,000 treatments, so a little less treatments, but still a high number of these treatments. So this represents, of course, a high number of drugs being handled by workers and to be also administered by workers. So we start in 2010, as I mentioned before, with a funded project. The funding was given by the Autoridade para as Condições do Trabalho, Portuguese of working conditions, the Authority for Working Conditions from Portugal, saying like that. The funding was given to the Public Health School and also from my institution at that time, Escola Superior de Tecnologia da Saúde Lisboa, and the aim was to evaluate the exposure to antineoplastic drugs in Portuguese hospitals and try to recognize the health effects related. We made the first sampling campaign between 2010 and 2014. We included 2 hospitals from the region of Lisbon. It was a little strange for us because this field work and also the samples analysis don't have any cost for the hospitals, but we were having some difficulties to engage hospitals in the study. So well, to accept to participate, that was good, to have an idea what can be the exposure in this setting to antineoplastic drugs. So we perform surface wipe sampling, and we also prepare, of course, reports that we send to the hospitals with the information about the contaminated surface and also the risk management measures that should be applied to avoid that contamination. So our idea was in the future to replicate this monitoring campaign and try to see if the hospitals apply some of the risk management measures, try to see if they have the impact needed to reduce the contamination. So after 2014, we concluded the study, but then because we published the results, several hospitals contact us to try to do the same kind of study. So I think the hospitals at that moment realized that probably they will have the same kind of problems that we identified in the 2 hospitals that were engaged in the study. So we were contacted by 4 hospitals that were located in other parts of the country. And we don't have funding for doing that, so the hospital supported some of the costs, mainly samples analysis and the transport. We follow the same approach to try to have an idea of the trend of the contamination, try to understand what was the situation in Porto. So what materials and methods we use at that time? We use the wipe sampling, as I described it. The sampling campaigns were developed in different days in each hospital. Before wiping, the gauzes were moistened with a specific solvent. The sampling was performed using a cover, try to define a specific area because the contamination was given by area units. The sampled surface were storage cabinets, preparation and packing benches, hoods, transfer chairs and beds for drug administration, worktops, infusion pumps and trays. Of course, there were other studies mentioned, for instance, the floor has a surface to sample. Well, we were not so much focused on the contamination, but more in exposure. So our main objective was to try to know the contamination of surface where the workers have to handle and touch. And with this, we have a more detailed idea what was the probability to be exposed to these antineoplastic drugs. So at that time, we use HPLC as a resource, analytical resource. Nowadays, and because we are still doing some of this work, because some of hospitals keep contacting us to do this work, we are now working with a partner from Italy, and we now have different capacity, lower limit of quantification, and we are now using a different analytical resource, and we are able not to measure only 3 drugs like we were doing at that time, cyclophosphamide, 5-fluorouracil and paclitaxel, but we are now measuring 23 antineoplastic drugs in the same sample. So the approach that we follow, we try to collect information of the number of chemotherapies prepared in that hospital, observe the tasks involving the handling of antineoplastic drugs and the pathway that drugs make before and after being prepared. Also, administering tasks are observed in detail, where the patients are being, normally they are seated or they are laid down in the bed, and they are nurses doing the administration of the drugs. We identify the surface most potentially contaminated and simultaneously most frequently handled or touched by the workers during their routine tasks. So this implies to stay several hours in the workplace and try to identify the surface that are more relevant for exposure and risk assessment. use the most applied antineoplastic drugs as surrogate markers for surface contamination. So until 2019, we use as surrogates the cyclophosphamide, 5-fluorouracil, and also the paclitaxel. Since 2019, we use as surrogate markers 23 different antineoplastic drugs because we have a partner, as I mentioned before. And of course, our idea is to measure all the antineoplastic drugs in the same sample. So what were our findings in 2014 and also after 2019? And what are the meaning of that result and the kind of risk management measures that we consider relevant to apply to reduce contamination and consequently exposure? So when we found, for instance, contamination found after surface cleaning and before antineoplastic drugs handling started, we can conclude that the cleaning process is not suitable, and of course has a specific risk management measure is that an adequate cleaning program should be defined, more focus on chemical contamination and worker safety, because mainly the main focus of the cleaning programs that are in place in the hospitals are focused on microbial contamination. Of course, it's an important aspect because we are dealing with immunocompromised patients, but all the kind of products that being used is to deal with microbial contamination that are not suitable for chemical contamination. And also the surface that are defined in the cleaning program to be clean with some frequency are surface that are not the surface handled and touched by the workers, but the ones that are more in contact with the patients. When we found contamination in most of the hospitals that we evaluate until now, we found contamination in surface that are not directly linked with the preparation and administration of antineoplastic drugs. So this is the meaning is that the contamination is spread in the hospital setting, in the hospital where they perform this kind of treatments, and probably there are wrong working procedures in place. So we believe that training and of course improving procedures is important to apply and is important to develop with some frequency and some regularly. So if there is someone new in the service, for instance, a new pharmacy technician, a new nurse, or even a new person responsible for the cleaning of the surface, they should have training, specific training, explaining all the procedures in place. Samples with the contamination by several drugs and with antineoplastic drugs not prepared and administered on the day of sampling, because we collect information about the drugs that were prepared and administered before sampling. And the meaning of this is the same, the spread of the contamination. Probably the surface cleaning protocol is not the adequate one. Probably the products are not suitable to remove the chemical contamination, and so on. So the same kind of risk management measures should be applied. Also, and particularly in the administration unit, we found a high number of surface contaminated. So we think, and we have discussed this with the Nurses Association, and we believe that there is a lower awareness about the safety procedures and also the risks that are implicated when handling this kind of drugs. So of course, training and improving of working procedures in this kind of specific context is very relevant, and the workers of course should be involved in a more frequent manner in the training and all the information that is being handled related with the administration of these drugs. Also, we found contamination in the patient's toilets, and this was something that we have done in a specific hospital. This toilet was used by the patients and their families, but because most of these patients, they have some limitations when to circulate, and they have some difficulties, so they are accompanied by family, not so much nowadays because of the COVID, but before they were all, normally they have one patient, one family member with them. So this WC, this toilet was used by the patients and by the family member. Normally all the services have different toilets for the workers and the patients. But what we noticed was that all the surface that we analyzed in this toilet was all the surface were with contamination. And the surface that we select was the ones that are always contact when people use the toilet. So there is also some kind of risk for the family members that are not suffering any kind of treatment that can be exposed also to these drugs. We also look for contamination inside and outside the protection gloves, and we found it. So probably also the frequency of substitution of gloves, the type of gloves and so on are not very suitable in some of the hospitals. So there are some actions that need to be developed to guarantee that the gloves are the adequate and they are being substituted in the frequency needed. So several samples with multiple contamination. This is something that we found in all hospitals. And this is saying that probably when exposure happens, also has a mixture. So workers are exposed to a mixture of antineoplastic drugs. Also, we found that the package that is coming from with drugs that is coming from the supplier is also bringing contamination. So we believe that the warehouse and logistics workers can be also exposed when contacting this package. So some kind of information should be given to these workers and inform them that the handle of the package should be done with adequate gloves. So how we think that the surfaces are being contaminated? Well, because workers are not so much aware that the package can be contaminated, has these pictures as I showed to you, and then they handle the package and then they handle some kind of computers and other kind of equipment that is being used by other workers. So this is a common way to contaminate surfaces that are being used by several professionals. Also in here, when the gloves are not removed after handle materials or equipment that can be contaminated, and then the same kind of material is being handled by people without gloves. And also the same way, the same gloves that deal with the preparation and administration tasks are also the same gloves that are being used to make registers and other kind of tasks that are not directly linked with antineoplastic drugs, but results in a cross-contamination of all the surface in the workplaces. So some of our main publications about our project, the idea was to raise attention for this issue, to give some information besides the reports that are also available in the Portuguese Authority for Working Conditions, was to give some awareness for the science public, or the public more related with science, with the need to develop more work about exposure and also the health effects related with the exposure to these drugs. So the most important conclusions until now, because we are still going on with some field work and also some reports to the hospitals, and we still want to give some inputs to the regulation about occupational exposure to antineoplastic drugs in EU, is that all the hospitals that we analyzed until now, more or less 7 hospitals, because we are now engaged in studying another hospital, but the 7 hospitals that we study in Portugal, all of them presented surface contaminated with antineoplastic drugs. So this is still a concern in Portuguese hospitals, and exposure can happen by dermal absorption. Preparation areas presented less contaminated surface than administration areas. So we noticed that probably the pharmacy technicians are more aware of the risks that implies the preparation and also the handle of these drugs than the nurses. So if we should try to identify what is the workers group that need more training and information, it's clearly the nurses. Need of constantly promote good practice and safety procedures among all the professionals that handle these drugs. And of course, we believe that it's important to monitoring in a routine manner, to have a monitoring program in place in each hospital to ensure that the application of safety procedures and to improve, of course, the risk management measures in place. So I think the monitoring program is something that each hospital should adopt and adapt to their reality. So what we think that is important to keep doing and to develop more research. Well, of course, we now have different analytical resources. We would like to have a Portuguese partner to work with us and not to do this collaboration with the Italian partner, because, well, they are our colleagues and of course, and some of them are our friends, but the Portuguese partner will for sure make it more or less expensive for the hospitals, because we don't have to deal with, for instance, the transport of samples for Italy. And we would like also to study other occupational settings where exposure to antineoplastic drugs can occur. We start studying veterinary clinics, and we study one of these clinics because in veterinary health, it's more and more use these antineoplastic drugs, and we found contamination. And also we identified that most of the clinics in the linked with animal health, they don't have in place any kind of risk management measure suitable to handle this kind of drugs. Also try to study more the home environment and try to understand how exposure can happen when most of the patients are doing the chemotherapy and in ambulatory manner. So what can be the family exposure in the home environment? And of course, there is a lot to do concerning risk communication to workers and of course also to general population, particularly family members and also the patients that go at home and in some way can put in risk their family members. So thank you so much for this time, also for your attention. I hope I can answer some questions of your interest. Thank you so much.
Thank you very much, Susana. So we can now open the discussion. You may raise your hands if you have any questions for any of the speakers of this session. We have already some hands on.
My question is for Professor Susana from the last presentation. And the question is just that as for us as consumers and patients, what can we do to improve or at least minimize the contamination, or if there's anything that we can actually do regarding that?
Hello. Well, thank you for the question. Well, I think there is a lot to do. Concerning workers' exposure, there are a lot of regulations already in place. Of course, we can include some other regulation and improve those already in place. And of course, we can try to support the hospitals' initiatives that are already in place concerning workers' protection. Concerning patients and also family, I think the information that is passed in each hospital differs. So it's no common information that in that way there is a lot to be done in Portugal, and not only in Portugal, because well, we have to adapt the message to our listeners. We have to adapt the fact. Yes, and for instance, a simple thing. Normally, we recommend in the hospitals that the patient doing chemotherapy use a different toilet than the rest of the family. Okay, that is important message. But if the home don't have two toilets, what is the message that should be transmitted in that way. So I think there is a lot to be done concerning how to interact with the people doing the treatment and how to inform the family, and mainly try to pass the message how people can be exposed so they can adapt in a proper manner the conditions and the recommendations that the hospitals normally do to the patients and their families. So I think there is a lot to do, and we hope also to give some inputs in that way.
Exactly, the information that you gave us is really important, and I guess that is the way to go, to bring awareness to the people that probably don't have much knowledge regarding science. I guess that's the way to go.
Yes, I think we have to translate science to be able to communicate in the proper manner, yes, for sure, and try to pass the most important information that people can decide for themselves in a more correct way. Yes, thank you. Thank you for the question.
Thank you so much.
Thank you, Margarita and Susana. Is there any other questions for any of the 4 speakers? Yes, we have here Ana Souza and Mayra. So maybe Ana, I'm sorry, I couldn't spot which of you raised your hand first. Let's start with Ana.
Okay, thank you. Mayra, do you want to do it first? No?
No need to. You can do it.
Okay. So thank you all for the wonderful presentations and communications. We definitely learned a lot. I have one question for Teresa that I would like to compliment, because she had a very difficult task to have half an hour to speak because Professor Arminda couldn't be here. So congratulations and thank you for accepting this and for doing this. Regarding the way that you calculate the predicted environmental concentrations, so in your formula you use the efficiency of the wastewater treatment plan removal technology in place, right?
Yes.
So is there a way that we can manipulate that part of the formula so we can try to predict our environmental concentrations based on the new technologies that we are developing?
That's actually a good question. That part of the work that I presented was actually done by Monica, Dr. Monica Santos. So I'm only presenting her work. I think it is possible to do that, but she didn't try it.
Yeah, okay,
to be sure. I can talk to her and then we will clarifies it better.
Okay, thank you. That would be good, because then we can also provide some valuable data that then the team from toxicology can use to understand better the concentrations after treatment, and also we can get some very important data on the toxicology aspect. As Cátia mentioned, we don't know so many things. We cannot calculate SSDs right now. So that's the work that we are developing with the team by Isabel.
Yeah.
They are trying to get us the numbers so we can use more species, so we can have a more accurate vision of what is going on.
The second work that I presented, I actually measured the concentrations outside the wastewater treatment plant, and we are expecting to measure even in the surface waters, but we don't have those results yet.
Okay, thank you.
Thank you.
Thank you, Ana and Teresa. So, Mara, you can now ask your question.
Hello. Thank you all for the presentations given in the last talk. They were quite interesting. And my question goes also to Teresa Gouveia, and like Ana Souza tell, congratulations for your talk. You have a higher challenge than the remaining ones. You were doing a longer presentation, replacing Professor Arminda, so thank you so much. You did a great job. And my question is, well, you show that there is one high-risk cytostatic drug that is the mycophenolic acid.
Yes.
The others also have problem, but this is the high-risk one. And my main question is this a main reason because it is highly recalcitrant to UV radiation, but you also have others that they are recalcitrant also, or this is a combined reason with this phenomenon of not being able to be treated together with the high consumption of this drug, and why it is so highly consumed, if this is the reason. I have no idea at all, and I'm just asking for curiosity. Is there any particular application, or in which type of cancer, or in which stage it is used? Could you tell us something about it?
Yes, mycophenolic acid is actually very used for organ transplantation, so it is not only used for cancer. Okay. And Portugal is actually known by good success in the organ transplantation surgeries. So we use a lot of mycophenolic acid, and it is the main reason why it is in high concentrations. And it is also very toxic, so the combination of these two variables makes it very toxic and of concern for the environment.
So it's a combination of the two effects. It's not being able to be degraded in an easily way, and because it's highly consumptive. But now, yes, I understood it.
We have already performed some assays of using mycophenolic acid and, for example, ozonation. And we are having some good results, so I think that we will be able to degrade it.
Perfect. Thank you so much. From my side, it's that.
Thank you, Teresa and Mara. Any further questions? I think we still have time for one more. Marcia.
Yes. Okay. So now I would like to thank all the speakers, because they were very nice presentations. And my question is for Susana Viegas. And it's about, do you have, is there any relationship between the contamination of the surfaces and the presence of antineoplastic drugs in the persons? I don't know.
Yes, I'm here, and I thank you for the question. Very interesting. Well, we tried the study in the beginning was also having a biomonitoring.
Yes.
Campaign involved. We were not able to do the biomonitoring biomarkers of exposure because it was implying 24 hours urine collection, and we tried to have it, but the health professionals were not willing to participate. So we have some difficulties there, but the previous studies, not the ones that we developed in Portugal, they found that not so much with the environmental exposure, but when they linked biomonitoring, biomarkers of exposure and biomarkers of effect genotoxicity tests, mainly micronucleus and comet assay, they found some connection between exposure and effects. And we indeed developed a part in a smaller group, the nurses linked with the pediatric treatment with antineoplastic drugs. We were able to engage those workers in a part of the study that we study we study the effect linked with antineoplastic drugs. For that, we use a control group because we don't have exposure data through biomonitoring. And we found out that those nurses have a big difference when we compare with the control group for the COMET and also to the micronucleus test. We have published those findings, and they really show that the exposure to antineoplastic drugs can really affect health, and they were linked with the findings that we have. And this is, unfortunately, is the trend in other studies developing also in Europe and the United States. And that's why the EU regulation is trying to include the hazardous drugs, where antineoplastic drugs are also included, in the directive that protect workers for carcinogenic and mutagenic agents. So the idea is to, well, not to have an occupational exposure limit, of course, but have something like it should be applied that hospitals should work for the lower contamination possible in the workplaces. So that is...
Or supply some kind of regular tests for the medical person just to check if they are exposed to high amounts.
Yes, health surveillance of workers is also performed in the hospitals, but is not specific for this kind of exposure. And there are no guidelines, so each doctor in the occupational health do this kind of surveillance in different ways. And sometimes the kind of health exams that are done are not linked with this specific exposure. So of course, the biomonitoring campaigns are important, also environmental ones, such as the wipe sampling, and also biomarkers of effects such as micronucleus and comet assay that nowadays are very well evaluated and really can link the exposure to effect, and also can identify the priorities concerning intervention in the workplace should be more used. Yes, I think we have to do a lot because yeah, the idea is that the treatment with this kind of drugs is going to increase. It's related with our higher expectancy of life, so we will have more and more people exposed to, and don't forget the home care service, in the elderly houses, don't forget also the veterinary health that is more and more using this kind of drugs without any kind of condition or any kind of specific resource to deal with these drugs. They also recommend that the pet is going with the family at home after treatment. So I think we have a lot to do concerning occupational and public health when we speak about this kind of drugs.
Thank you very much.
Thank you.
Thank you. Well, we are now 30 minutes after the time to end this session. Let me just check if there is some more raised hands. Maybe we can do one more question and then we need to go for lunch. Well, it seems that we don't have no further questions, at least at the moment, but of course this discussion can then proceed aside in the chat, for example, with the speakers. So I would like to acknowledge again and thank you to all the speakers for their availability to be in this session, for their presentations, and also for this fruitful discussion that was very interesting. I don't know if the organization wants to give some information for the participants regarding the return in the afternoon.
Okay. Yes, Isabel, thank you. So we will return after lunch. That was supposed to be very good in Melia Hotel. We have to wait until next year. If we want to talk, we can use the chat or we can just email each other and start some fruitful discussions, we hope. And we will see each other again at 2:30 for the first session in the afternoon. Thank you all and see you later. And obrigada, Isabel.